Well, very good evening to all of you, and good morning and good afternoon to all others who are joining from rest part of the world. Today, our guest speaker for the day is Dr. Samir Parekh from New York, and he's going to lecture us on At the Cusp of a Cure in Myeloma, Insights into the Pathogenesis, Modeling and Therapeutics. This webinar, as usual, is brought to you by Mumbai Hematology Group, supported by Jensen, Unipath, Lucien, and Nirvid Life Sciences, and very well managed by Perfect Square. I thank Team Jensen, Unipath, Lucien, and Nirvin, Mr. Yash, Mr. Kalpesh, and the team from Perfect Square, Executive Committee of Mumbai Hematology Group. Our guest speaker for the day, Dr. Samir Parekh from New York. All our discussants were themselves very eminent hematologists as well as medical oncologists. And you participants for sparing your Saturday evening, morning, or afternoon for whichever part of the world you're logged in. Tomorrow, that is Sunday, 29th of June 2025, from 11th a.m. Indian Standard Time, we have Dr. Reena Nair speaking to us on clinically relevant classifications of lymphomas. Let me introduce our discussions for the day, and we have a galaxy of them here displayed in alphabetical order. I will pause for a while for you to have a look on all our discussions for the day. They are displayed here in alphabetical order. Some more. So these are our discussions for the day. Now it's a time to introduce our guest speaker for the day. We have no other than Dr. Samir Parekh from New York. Let me introduce Dr. Samir Parekh to all of you. Dr. Samir Parekh completed his clinical training in hematology and oncology, followed by postdoctoral training in Dr. Ari Melnick's lab at the Albert Einstein College of Medicine. He currently serves as a director of the Multiple Myeloma Center of Excellence at Mount Sinai and is a professor of hematology, oncology, and oncological sciences at the Icahn School of Medicine at Mount Sinai. His research focuses on the pathogenesis of hematological malignancies and the development of individualized precision medicine therapeutics and therapies for these diseases. His lab uses an integrated system biology approach to study genome-wide methylation, gene expression, and DNA sequence variation to understand the pathogenesis and develop biomarkers and guide personalized therapy in B-cell malignancies, particularly multiple myeloma and non-Hodgkin's lymphoma. Today he's with us and he's going to lecture us on At the Cusp of a Cure in Myeloma, Insights into the Pathogenesis, Modeling, and Therapeutics. Well, thank you so much. I stop over here. Over to you, Dr. Samir Parekh.
Thank you so much, Amit, Dr. Agarwal, and all the organizers. I'm really privileged to give this talk to all of you that have taken time out from your busy schedules with patients and family. I used to attend Dr. Agarwal's lectures as a medical student in Bombay, and I'm really happy to be able to turn full circle and actually give this talk at your seminar series. And I see even my father, Dr. Sunil Parekh, has joined and is able to attend. So this is really a nice opportunity to have a wonderful interaction. So as Amit said, I lead the multiple myeloma group at Mount Sinai, which is a major center on Upper East Side of Manhattan. And in some of my other roles, I also am a chair of cancer biology. So you will see some of the basic science incorporated into the talk today of how we are moving the field and treatments forward towards a cure in myeloma. The Cancer Clinical Investigation Program involves approximately 50 different scientists as well as clinicians that all come together, and we work to develop unique clinical trials that can actually go back to patients and have led to several FDA-approved therapies. So it's been an exciting time, and as you all know, myeloma treatment landscape has changed tremendously. So I would love to share that excitement with you. So this slide shows where our center is located and how we work generally. The main campus is on the Upper East Side of Manhattan, but as you can see, we have different sites in downtown Chelsea, Brooklyn, Long Island, and New Jersey. And we actually see patients from all over tri-state area, US, and even rest of the world. Our model has always been to try to give care locally, and patients come to us when they particularly need more advanced care, like clinical trials or CAR T or others. But we really believe, because of the long-term nature of myeloma treatment, that local care is the best care. And we've had several patients from India as well who come back and forth, and we've maintained that similar model. When I joined Mount Sinai, um, little over 10 years ago, there was a revolution in genomics in our institution where a mathematician, Eric Schad, had hired close to 300 different faculty from Silicon Valley and Stanford and other great universities to try to help different disease groups understand and treat um, diseases and cancers in a more data-driven way. And we collaborated particularly with Professor Joel Dudley, who had developed unique models for using RNA and multimodal sequencing to actually classify disease and treat disease. And we were able to publish these initial observations in a number of papers where we actually found that myeloma is not just the three types by cytogenetics that we classically understood, but is much more heterogeneous. And we found 12 groups as we could combine different genetic lesions that co-occur. And each of these groups had a different prognostic impact and also different therapies associated with each group. So, with this rationale in mind, we conducted a pilot clinical trial where we used next generation sequencing of both DNA and RNA. And this allowed us to salvage patients that were very late stage, relapsed after 5 or more lines of treatment, and had literally no FDA approved treatment options. What was surprising in this study, you can see this waterfall plot. Majority of the patients showed a decrease in disease burden, but in large part, this was based on RNA-based findings that we used from their sequencing. Now, at that point, there were commercial agencies like Foundation Medicine and others that were doing DNA sequencing, but the RNA-based sequencing was largely an open area, and people would not really use that for drug repurposing. And this study showed the feasibility of doing that and also efficacy in a very late stage myeloma patient population. And on the basis of that, we got our NCI R01 to investigate and develop this further, even for patients now that are getting treatments and undergoing new therapies like CAR T and bispecific antibodies. So we started giving the initial CAR Ts on clinical trial about 6 or 7 years ago, and also were involved in the seminal trials that led to approval of several bispecific antibodies and now several novel IMiDs like iberdomide and mezigdomide. In fact, we are a reference center for understanding the direct effects of the IMiDs in the bone marrow and have an extension collaboration with Oxford University to do this across the world. More recently, many of you may have seen Dr. Jagannath's presentation at JCO, at ASCO, EHA, and published in JCO, where he described the long-term follow-up from not just our patients, but all the patients that received uh, the J&J CAR-T, CARVYKTI on the initial clinical trial. And as you can see in this curve, almost a third of the patients look like they have gone into stringent complete remission. In fact, at our site, we have data that shows that they are continuously MRD negative for 5 years. And this is really important because we have analyzed patients that have this pattern of repeated MRD negativity for 5 years. This correlates to a very good long-term overall survival. These patients go on for 10, 15, even 20 years with very little relapse at that point. So this really gives us hope that we finally have a chance of giving long-term MRD negative remissions in patients even with advanced disease. On the basis of this, we are now testing CAR T even in frontline settings. We enrolled the first patient in the US on CARVYKTI in an upfront clinical trial, and this will read out over time, and we will update to you. As an example of this, this is a patient in my clinic who is the longest living survivor and after CAR T, and she is now 6.5 years out of CAR T, doing well, has no side effects. In fact, flies between Florida, where she spends half the year, and New York, where she spends half the year. She is with her husband, who's a gastroenterologist, and they both really have amazing quality of life. This is their photograph on their 50th wedding anniversary. So we have many such patients now that have crossed 5 year mark and are actually understanding their T cell changes are quite different compared to the patients that have conventional course with CAR T. And we can talk about that. So the reason we wrote this editorial and seminars in hematology and actually the entire issue with different people at Mount Sinai was this paradigm of using MRD detection in a sequential way to really identify patients that we could be more confident that were cured. And as you all know, as practicing hematologists, that there's only the tip of the iceberg that we detect with serum markers like M-spike. And once we get to undetectable, we have to use MRD measurements by either flow or next generation sequencing. At our center, we typically use next generation sequencing with the clonoSEQ assay. In about 10% of the patients, we may still use flow if they are not able to capture a signal with clonoSEQ. But the use of MRD has been now approved as an earlier uh, measure for outcome and success of clinical trials. The US FDA unanimously voted last year that we could actually start using MRD as an endpoint. I think this is really transformatory in the field because the earlier readout will help us develop better treatments. However, it is still an open question of whether a single or even two readings or more readings are needed to say what is the significance of MRD negativity. So we did a study looking at almost 1,700 patients with myeloma that were treated at our center. And we have a protocol that if patients are in complete remission, we do sequential MRD testing. This helps us identify a cohort of patients. In the paper we published earlier this year with Dr. Chen, we had 76, but actually our overall cohort is about twice that size. As you can see in this pretty remarkable overall survival, 100% of the patients that were MRD negative at 5 years were alive. 95% were myeloma free at 6 years, and about 88 at 9 years. And most importantly, 70% were off all myeloma treatment. So we are using this in our practice as a decision-making tool to help counsel patients. If you've reached 5 years of maintenance, it may be okay to start thinking of stopping. And each patient is individual. Some feel more comfortable keeping the maintenance on. Some people prefer to stop it. But we can at least tell them with data that your chance of relapse is under 10%. And I think this is the way MRD can be really used in the future. The next question that we are trying to address is why some patients achieve long-term MRD negativity and some don't. And this really has to do with a very complex interaction between the myeloma cells and the T cells and the microenvironment, as I'll outline in the next few slides. What is interesting is that the type of CAR T being used doesn't seem to matter that much. So these were the seminal papers that described the Abecma CAR T product from BMS and the Carvykti CAR T product from Janssen and their overall survival. And you can see that with Abecma, the overall survival and PFS seems to be inferior to the Carvykti, which had median PFS of about 31 months. But there is a center-specific effect. If we look specifically at the patients treated at Mount Sinai, and we have now much longer follow-up than either of these two published studies going 6 years and out, our overall survival in our patients is about 60% for either CAR T. This suggests that it's not the type of CAR T, but the care the patients can get even after CAR T. As patients relapsed from Abecma, for example, we were able to sequence them with other treatments that kept them alive, and they still remain to be alive. So the ability to think of life after CAR T and how you are going to manage patients is extremely important. A brief mention about a third CAR T that is looking extremely promising, called anito-cel, shows that it has very high PFS rates and OS rates, but importantly, none of the patients in the cohort that got anito-cel had delayed or severe neurotoxicity. And this is a very promising signal, which was presented by Gurbaksh Kaur from our group at EHA this year. We will continue to follow with interest this clinical trial. Maybe this has the balance of efficacy and reduced toxicity that can really serve as a good middle ground for a lot of the patients getting CAR T. But it remains to be seen. What do we do when patients fail CAR T? We've actually developed an extensive genomics guided platform to analyze these patients in real time. Platform is called RADAR, And you can see here it's a pretty complicated bioinformatic assessment of both the DNA and RNA. But overall, what we end up doing is creating a report that can actually help guide patients and tell us what would be a good next line option. So this is what the report looks like, and we can actually not just identify the genomic landscape and also the recurrence scores and things like that. We also look at expression of BCMA and GPRC and mutations in these things. This is one patient who had developed a mutation in GPRC5D in real time, and we could actually model it and show that this mutation would prevent the activity of the bispecific. We are able to then use this information to guide next lines of treatment as we have multiple GPRC based CAR Ts now, as well as BCMA based therapeutics, including ADCs and others, that we can then, in a thoughtful, rational way, guide patients towards. Another useful nature of this sort of sequencing is that we identify mutations that can be targeted therapeutically by drugs that are outside the myeloma toolbox. For example, this patient was an elderly physician who had gone through CAR T and was relapsing and ultimately was going to hospice when Dr. Jagannath called me and said, do we have any sequencing information that could potentially help the patient? And based on this sequencing, we identified a BRAF V600E mutation, which, as many of you all know, is usually seen in melanoma. And collaborating with a melanoma lab at Sinai, we came up with a combination of drugs that would very systematically block MAP kinase signaling. And you can see the impact of this was the patient's light chains dramatically reduced. All his extramedullary disease in the skin went away, and he went on to live for 2.5 years, even got bispecific antibodies after that. So really, this is how we employ real-time sequencing in our patients. A side effect of doing this sequencing, since it's also looking at germline mutations as well as mutations in the tumor DNA, is that we have now identified in about 8% of myeloma patients mutations in well-known cancer drivers in the germline. These are associated with familial cancers, genes like BRCA1 and 2, TP53, and several genes associated with colon cancer. So this sort of finding has implications for young patients, for patients with a family history of any heme malignancy, but particularly myeloma or MGUS, or other cancers. And we have now created a separate pathway for these patients. This is led by a young faculty in my group called Santiago Thibot, where they screen and counsel not just patients but their families, because these germline mutations have implications for the children of patients with myeloma with these mutations, and they can get early screening and appropriate counseling to prevent malignancies in the family. Our program now is mostly focusing on developed treatments for post-CAR T settings and for improving the efficacy of CAR Ts and bispecifics. And we've had several publications in this regard. I'm going to bring you all an update on what we are doing. There are several groups around the world that have published that there's BCMA antigen loss in CAR T. Here, in an elegant study by Nizar Baylis, he showed that there was, at the single cell level, loss of BCMA, as you can see in the single cell plot, and flow cytometry. This happens more in bispecifics than it does in CAR T. In CAR T, it happens about 5% of the time. In bispecifics, we think it happens between 20 to 30, even sometimes more percent of the patients, but it varies from the bispecific to bispecific. So in this study, he actually showed that certain bispecifics lose binding if they have mutations in BCMA hotspots, but you can actually have some that maintain binding, and that way you can really personalize the therapy if you know what the mutation is. We have gone on to study both bispecific and CAR T patients with mass cytometry and single cell RNA sequencing using a cutting edge methodology called CytSeq, which simultaneously analyzes both protein and mRNA from individual cells in the bone marrow and peripheral blood. And computationally, we can then project back which cells the sequences are coming from. And this really helps us understand the disease and resistance mechanisms and high resolution. So I will show you some data from this that is helping us understand and develop new treatments. Um, this is from CAR T patients, and you can see a very clear pattern here where patients that had four outcomes marked in red under VGPR had reduced expansion of CD8 CAR Ts as compared to patients that had good outcomes. You see this expansion of CD8 CAR Ts. This is intuitive. If the CAR T doesn't expand, you're not going to get a response. And this happens more frequently than we would like. Particularly, there was a period when we were getting out of spec CAR Ts, which thankfully is being seen less and less now. But CAR T expansion needs to be tested. It's not routinely checked in commercial labs, so we do it in the research basis, and we can see this. How can we mitigate this phenomena? So I had mentioned earlier we had a large collaboration with Oxford University looking at the bone marrow of patients treated with new IMiDs like iberdomide. And this study was published recently, showed for the first time that the novel cell mods can actually activate T cells and NK cells in the bone marrow and does so dramatically better than the previous generation of IMiDs. So we therefore had an idea that we could use these new agents to promote CAR T. And in fact, when we were able to test this in vitro and in vivo, we are seeing a synergistic effect when we add iberdomide both during CAR T production and post CAR T as a maintenance. It improves the cell kill, persistence, cytokine production. So we have a clinical trial now being developed to help it even for priming before the patients go into CAR T. One of the other insights that came out of this effort was a remarkable finding about the CAR T microenvironment. This is one of the hottest topics in CAR T biology. What are the factors in the microenvironment that are inhibiting CAR T? And what we noticed a month into the CAR T, when the CAR Ts actually are going away, patients had increased levels of myeloid suppressor cells in their bone marrow. And these myeloid suppressor cells had a very high level of a gene called S100A8 and A9. This is actually a gene that is an alarmin. Way our body uses this gene is a regulatory mechanism to switch off when there's too much inflammation. The CAR Ts release cytokines, they cause inflammation, but when there's an excess, this is released to actually dampen the T cells. It is well known in rheumatology and other fields. And we were reassured as another group led by Dr. Madhav Dodapkar that looked at a CAR T made in UPenn found exactly the same findings that patients that were doing poorly had a myeloid population that had high levels of S100, A8, and A9. So these proteins called alarmins are part of the DAMP family, damage-associated molecular pattern family, which are released in response to tissue injury anywhere in the body. They can be secreted by myeloid cells and neutrophils, and also tumor cells in MDS, AML, and multiple myeloma. This elevation actually recapitulates MDS in an elegant transgenic mouse model that was published by Alan List many years ago. And it does so by inhibiting T cells and altering the Treg or regulatory T cell balance between um, the bone marrow and peripheral blood. What we found looking at our cohort of CAR T patients was that the patients that had very poor survival versus patients that went on to do amazingly well many years out of their CAR T had an entirely different pattern of S100 in the peripheral blood. This served as a biomarker for distinguishing very early on whether these patients were going to have good responses or not. And the reason we think this is important is that we actually saw this correlate very well with increased regulatory T cells, including myeloid suppressor cells, and very, very low CAR Ts. So this raised the hypothesis, are these gene products directly impairing CAR T? And we set up assays in the lab using homegrown CAR Ts and target cells that express BCMA to test this hypothesis. And what we found was as we titrated increasing levels of this S100, A8, A9 alarmin, there was a dose-dependent decrease in cytotoxicity by the CAR Ts. So there was a direct effect of this protein in inhibiting CAR T killing. And in a serendipitous manner, I happened to run into Dr. Amit Verma, a friend and colleague who works at Einstein on MDS, and he had been developing therapeutics against this protein. And in a wonderful collaboration that ensued thereafter, we have actually tested these in our models of myeloma CAR T, and we can show that when the S100 is blocking the CAR T function, if we use the inhibitors or monoclonal antibodies against S100, A8, A9, we are completely able to rescue the CAR T function. This is very exciting and something we are hoping to take forward to clinical trials. What about bispecific antibodies? A similar phenomenon exists there where patients that have very good outcomes after bispecifics have low levels of this alarmin, whereas patients that don't have good outcomes have much higher levels, and it's independent of what bispecific you look at. In the lab, we have demonstrated that titrating this in the bispecific context also reduces bispecific induced killing of myeloma, and we can rescue that by inhibiting it with the therapeutic antibody. We have now taken this in vivo. This is a previous experiment that was done by a group from Baylor looking at a particular reagent that could kill MDSCs that was secreting S100, but we have now reproduced in different mouse models with the help of Spanish collaborators. This works even in vivo. Another challenging setting where patients present is with extramedullary disease, that is disease outside the bone marrow. And as you can see here, Darren Pan, one of our fellows now at UCSF, showed that the survival of patients after CAR T was significantly worse in this population. And this has been shown across the world in different clinical trials. On the right is a meta-analysis of this. So what can we do about this? Well, the first thing we started doing was by biopsying these extramedullary lesions. And this gave us the first clue. And we used a very cutting edge technology called spatial transcriptomics to analyze these biopsies. On the right is an extramedullary lesion. On the left is a bone marrow from the same patient that had progressed after getting CAR T and bispecific. You can see here on the bottom, already there are some clues of why this patient may not be responding. You can see there are no yellow cells over here, whereas here you can see these yellow cells, which are CD3 positive T cells. So the lack of T cells really will inhibit response. You can imagine in a bispecific, if there are no T cells, no amount of bispecific is going to overcome that. Similarly, we can now plot at an individual cell resolution in situ what is going on. And you can see this big bundle of cells in the extramedullary lesion on the right is mostly filled chock-a-block with tumor cells. And this is a heat map of all those cells. If you follow just a little bit more with me, we were looking for evidence of heterogeneity even within this tumor block, and we really found a startling difference. There were some areas, like the area over here, that had some myeloid cells in it. And in fact, that area did have CD8 T cells. So there is heterogeneity where you have myeloid antigen presenting cells in some areas that can then attract T cells. How can we therapeutically make use of this? So there are ongoing clinical trials with injections of FLT3 ligand, which is a drug similar to GCSF, but aimed more at dendritic cells. And this can stimulate dendritic cells in these deserts of myeloma cells to actually attract T cells and cause T cells to do their job. We have shown with the latest spatial transcriptomics technologies that our findings around S100 inhibiting T cells can be validated even in situ. As you can see here, the S100 expressing cells causes exhaustion as measured by the marker TOX. And when the cells don't have S100 in them, the neighboring T cells are actually doing quite okay. So this really gives us hope to improve on the efficacy of CAR T and bispecific, even in challenging patients that have extramedullary disease with novel therapeutics like the S100 bispecific or monoclonal blocking antibody. What about sequencing CAR-Ts and bispecifics? So we have started analyzing this, as have other groups, many years ago, and we published in Blood our initial results, which we had pooled data from both our institution as well as Memorial Sloan Kettering with the help of Sham Melanchody. And what we found after 140 patients treated with CAR-T were that 79 patients could actually go on to a second treatment, and the majority of them had pretty good overall survival. So, what were the treatments that seemed to work in that setting? The three treatments that seemed to work were if they got autologous stem cell transplant or if they got salvage with a second bispecific antibody or CAR T. If the patients got conventional drugs like doublets or triplets, their PFS was dismal in the range of 3 to 4 months. So really the message here is after CAR T, you're much better off getting a bispecific or a salvage transplant before you go on to other treatments. The reason for this, we found in doing deeper analysis of the patients, is that patients' endogenous T cells after CAR T are still capable of being activated by bispecifics. And I'm showing you here an example of a patient that got second bispecific. And you can see that there is robust activation. These are different activation markers on CD4 and CD8 cells. And all of them went up despite the patient being post CAR T. We have now published on this data, and we can show that after bispecific salvage as well, people can go on to second bispecific or even CAR T, and this is also quite effective. And this work was led by Tariq Mohideen, who's a fellow at Mount Sinai and now going on to Dana-Farber as faculty. What we did find is that activation markers, and in fact, this is relevant for subsequent treatments that incorporate exhaustion or anti-exhaustion measures, increase after sequential bispecific treatment, particularly the patients that seem to not respond have an increased expression of exhaustion markers like PD-1 and TIGIT, and these may actually be treated with anti-PD-1 or TIGIT and be rescued. So we have now modeled this, and we have clinical trial that will be started based on these findings. I had presented this in an ASCO education session last year, and I thought this would be a useful summary for the audience in the Bombay Hematology group to highlight differences between sequencing after CAR T and bispecific. So if you'll bear with me, I'll walk you through the differences. In CAR T, the process involves in vivo CAR T expansion, and there is a long period before the patient relapses that can range from 12 to 36 months. When the patient relapses, BCMA is usually still expressed. Therefore, a second treatment can be used. However, we recommend switching to a different BCMA CAR T because reinfusion of the same product usually doesn't work because the patient develops autoantibodies to that product. We can also time the CAR T. The longer the interval between that CAR T and the second CAR T, the better the outcomes are. If the patient is cytopenic after CAR T, autologous transplant or a boost is a salvage option because it will help you increase the counts of the patients. And because there's less T cell exhaustion in these patients, bispecific antibody salvage works well, which I showed you just a few minutes ago. In contrast, after bispecific therapy, where there's constant exposure to the drug, when the patient progresses, usually the PFS is roughly 12 months for most of the approved bispecifics. The relapse occurs on treatment, and there is more antigen loss here, up to 30%, because there's constant selection pressure by the bispecific antibody. The options at that time are that we could either switch to a different target. So if the patient got BCMA bispecific, we give them GPRC or vice versa. And CAR T is also feasible, particularly if BCMA is still expressed and not mutated. So we can check this. Poor man's version of this check is by looking at soluble BCMA, which is a quick ELISA test. Autologous transplant is very important in this setting if the patient is cytopenic, similar to CAR T, because it can bridge the patient to other therapies while debulking him or her. And as I showed you with the S100 story, the immune microenvironment is very important. Regulatory T cells have been shown to induce resistance. And in fact, one of the drugs that is known to overcome this is already available, daratumumab. It reduces regulatory T cells expressing CD38. In the last part of my talk, I'm going to focus on two or three side effects that we see all the time and how we are thinking about mitigating this. One of the side effects that we published on very early on in Nature Medicine, was a very unusual side effect where patients, about 5% of the time, can develop a progressive um, phenomenon that resembles Parkinsonism. And this generally happens when there is CAR T overshoot. Patients go into complete remission, as this patient did, and they can come to your clinic with a variety of different features, mask-like faces, tremors, postural instability, and these patients usually have a progressive course that gets worse. This is because the CAR T is going into the brain in particular areas and causing damage, as you can see with this pre and post scan showing loss of uptake. We were actually able to prove that those areas of the brains have neurons that express BCMA. And actually, on the left, you see the brain gets completely fibrosed with the CAR T infiltrate, and this can almost become irreversible. This is because some of the CAR Ts have survival genes like IL-7 receptor in higher amounts, and this causes persistence. Um, we have now ways to mitigate this using a variety of different methods. People have tried to kill off the CAR with cyclophosphamide, intrathecal therapy, anakinra, and others. But I'll talk to you about a new way of approaching this in a little bit. The second side effect is infection. And early in the experience, we had patients that had gotten CAR T even die of COVID and other infections. Here was a patient that was in complete remission, in fact. And after the CAR T, he wanted to bulk up to impress his girlfriend. So he went to the gym. Unfortunately, got COVID and passed away. And this set up an entire program in our institution to analyze what had happened, how could we mitigate this. So we looked at over 400 patients, and with the help of our microbiologists like Viviana Simon, we had a series of high impact papers that showed the patients that were most vulnerable were patients that were on either anti-CD38 therapy or BCMA targeted therapy or both. And this remains true even in the post-pandemic area. In fact, they have decreased T cell function, and this can be a useful way of screening bispecifics because we can show even with the latest vaccines, there is a decrease in the ability to neutralize variants like Omicron and others. And we are now using flow cytometry as a way of figuring out if these patients have more susceptibility to infection. Of course, everybody gets IVIG, but we can space out the bispecific more in patients that are extremely lymphopenic or more prone for infection. What about cytokine release syndrome? Everybody uses tocilizumab, but we also showed that because different CAR T products have different patterns of CRS, we actually need to individualize even CRS management. And using an extensive um, platform of cytokine measurements, we were able to start using anakinra, which is a blocker for IL-1 related CRS, and it works extremely well in patients that are refractory to tocilizumab. We had published, along with our immunology colleagues, using a chip-based methodology that can check several cytokines within a couple of hours. This is extremely useful in the clinic and can be easily deployed. Another thing that we are deploying are remote wearables. These are trackers similar to your Apple Watches that continuously track the temperature. And as you know, CRS is usually manifested by an increase in temperature. But by continuous tracking, both by wearables and now we are integrating even special cytokine measurements, we have found we can get a lead time in most of these patients that includes that improves our CRS detection by several hours to several days. And this can completely transform the paradigm of how we deliver CAR T, which is mostly inpatient, to an outpatient paradigm and do it safely. With the help of this, you can see the change in the early warning that we can get in these patients. And we have different metrics that we use of how much the temperature goes up, how sustained it is, and things like that to have confidence that patient is going to get CRS. In the last minute or two, let me talk about a publication that is under review at New England Journal from our group. This is a new side effect that is coming up after CAR T. It's extremely rare. We had a patient developed a facial rash um, several months after CAR T. Even though he was in complete remission, he had lymphadenopathy and, in fact, leukemic disease with very high lymphocyte count. And this was because he had developed a T cell lymphoma from the CAR T. And you can see the histology of the T cell lymphoma was unusual. It was both CD3 and CD8, CD3 positive, but CD4, CD8 negative, so double negative phenotype. It was clonal in the skin, bone marrow, and blood. And we were quite um, reluctant to use the standard therapy for lymphoma on him, as our collaborators in Australia, who had also had a similar case, had actually treated patients with allogeneic transplant without much efficacy. So we devised a personalized therapy for him using a screen that looks at what drugs kills just the CAR T or the lymphoma and spares normal T cells. And among several top hits in that screen, we found actually old chemo drug, daunorubicin, did quite well. Um, and we found another clue here by expression of chemokine receptor 4. We could actually use mogamulizumab, which is a drug used in ATLL or another type of T cell lymphoma. And this combination worked very well for the patient. He is now in complete remission over a year, year and a half, and his endogenous T cell counts have actually recovered. So we are using this sort of screen now to actually switch off the CAR T in different settings. And I was just told a couple of days ago, one of my colleagues at MD Anderson has used carfilzomib based on our screen on a patient that was getting severe cytopenias after CAR T. And the CAR T numbers are dropping even after a few doses. So with that, just a word on our existing plans and collaborations. We received recently a large multi-center grant to work with leading experts from around um, the US, including folks from Stanford, UCSF, Einstein, and Hackensack, and others. Um, each of these individuals are experts in their field of making better CAR Ts, doing integrated genomic analysis, functionally understanding what are the genes responsible for high risk disease and how can we overcome that? Because I think that's the next frontier to improve on. Our overall paradigm was published in Science a couple of years ago, where we take the complexity of pathogenesis and address it with advances in technology and treatment to create a personal paradigm for each patient and help get them to a cure. And this really takes a village. I have to thank Dr. Jagannath, who is the founder of our program, and all the attendings that help in the clinical work and trials. Also want to thank the lab group and the clinical staff, our collaborators, and all our funding sources. So with that, I'll stop and take questions. Thank you all.
Thank you, Dr. Samir Parekh, for a wonderful talk. That was wonderful, fantastic, sir. Thank you, sir. And now I request all our discussants to please put the hand raise signs so that we can start with the question and session with Dr. Samir Parekh. Yes, Dr. Chepsy. your question.
Thank you, Dr. Parekh. There was so many things that you mentioned in your talk, so a lot of things to follow. But just one thing that I wanted to ask you was you had spent some time on the sequencing of therapies post CAR T and the bispecifics. So, if I gather right, so you are suggesting that in case you're using a bispecific, you can reserve your auto for a relapse. Is that what you're suggesting? Should we wait for a relapse to do an auto, or is there a benefit of doing an auto just immediately after your bispecific without a relapse? That's my question.
Right. So we usually don't do the auto without a relapse. We do it if the patient relapses with cytopenia, because the limiting factor in making CAR Ts or even doing a second bispecific is that you need to have sufficient lymphocytes to harvest and make a CAR T. If these patients are so cytopenic that there are no lymphocytes to harvest or act when you give the second bispecific, that's when the auto comes into play. And I would also distinguish this auto is different from the upfront auto that many patients get. That still has a role. I think that until CAR T gets more widespread and replaces the upfront autos, they can still get the upfront auto, but post bispecific, please reserve the second auto for cytopenic patients and upon relapse.
Thank you.
Thank you, Dr. Chepsey. So, anybody has any other question? I think that was a fantastic lecture, sir.
Thank you. I see Dr. Agarwal's here. Thank you, Dr. Agarwal.
Yeah.
Can I ask one more?
Yeah. Please go ahead. You have a lot of time.
So Dr. Parekh, once again, so in this
Great.
under revision article that you are thinking about, like the MD Anderson team chose Carfil, whereas you didn't choose that. How do you make a choice between the drugs which you get out of those signatures?
Right. So the two scenarios were slightly different. The patient that we were treating had developed T-cell lymphoma. So we wanted to give the patient drugs that would work or at least be more likely to work in a T-cell lymphoma setting. That's why the mogamulizumab and anthracycline were helpful because gemcitabine and anthracycline is a acceptable T-cell lymphoma therapy. We thought we would start with that first, and if the patient relapses, give carfilzomib upon relapse. Thankfully, we have not yet seen relapse in the patient, so we are reserving it for second line. In contrast, the MD Anderson patient, right off the bat after the CAR T, unfortunately developed complications like GI colitis and HLH, which was biopsy proven. This was not responding to standard treatments, including steroids, cyclophosphamide, anakinra, and others. And that's when patient and family approached us saying, please, can you help? We spoke to the doctors and told them about the screen, saying that carfilzomib, even low dose carfilzomib, which was what we had tested in the screen, may be able to help. And that's how the patient got carfilzomib.
Thank you. Thank you for this wonderful work you're doing.
Thank you, Dr. Chepsey. Dr.
Dr.
Ajit.
Goel has
Dr.
a
Ajit,
question.
please go ahead.
Thank you, sir, for such a wonderful and insightful talk. So your clarity in breaking down the multiple pathways is really appreciated. So my question is, with all these new treatment strategies gaining momentum, do you think we are ready to redefine treatment endpoints in myeloma, like from complete remission to sustained MRD negativity, especially in transplant ineligible patients?
So Very good question. I'm going to break it down into two parts. I think that we are ready to redefine endpoints, and I think sustained MRD is a very good endpoint. The duration of sustained MRD, however, still needs to be better defined because the early studies looked at it for 1 year having sustained MRD. That's not enough. We have gone and looked at 2 years and even 3 years. There is a drop off of patients that continue to relapse. So hence, my initial slides that showed 5 years sustained MRD negativity had the lowest drop off under 10%. And I think that's what really starts then changing the way we think about cure. The
Mm.
second part of your question was aimed at transplant ineligible patients. While our older patients into their 80s and 90s are now getting bispecifics, we have to remember the goal of treatment in these patients is still quality of life. Merely getting them to MRD negative and sustaining them may not necessarily be important if their quality of life is not great, particularly with new bispecifics like anti-GPRC5D. Patients get skin peeling, difficulty swallowing, infections. So keeping all that in mind, we titrate our treatment intensity and frequency to give the patients best quality of life, especially in the geriatric population.
Thank you, sir. Any other
Thank you.
question? Yeah. Dr. Goel? Fine. Dr. Gurpreet Kaur, your question, please.
Good evening, sir. Thank you for a wonderful lecture, sir. My question is with regards to monitoring of these patients on CAR T therapy, sir, like with regards to the numbers of the CAR T cells or the copy numbers, how often do you do it and how do you do it, sir, in your lab?
Yeah, that's a very good question. Um, our CAR-T group has 3 different attendings, and we have a total of 10 faculty. And if you ask each of them how they do it, they will give you a different answer. So we are trying to standardize this for our program, where we at least do, at the bare minimum, a PET scan and bone marrow and examine patient for clonal hematopoiesis or chip-related mutations post-CAR T at 6 months, 1 year, and then annually thereafter. These patients are maintained on IVIG and receive an extensive post-CAR T vaccination similar to the auto or allo patients. These are most important for their monitoring. The monitoring of CAR T levels and cytokine levels comes into play when the patients have any of the rare complications I mentioned, including neurotoxicity, including cytotoxicity, or GI colitis. And if you suspect this is happening, that's when involving a research lab, getting the CAR T measured. Sometimes we even have to measure it in the lumbar puncture fluid and looking at the cytokines and then devising an appropriate mitigation strategy early becomes very important before the process becomes irreversible.
Thank you, sir.
Thank you. Thank you, Dr. Gurpreet Kaur. So there is one question which has come from the audience. Dr.
Yes.
Yvonne Tent has a question. In a patient who is a relapsed multiple myeloma and unfit for second transplant, in view of financial constraint, how long should we go on giving daratumumab to this patient?
That's a lovely question. I think that our practice has been to give daratumumab for at least 2 years, along with an IMiD, to maintain these patients. After that, we can continue the IMID alone if the patient is in stringent complete CR and stop the daratumumab. This also reduces infectious complications, need for IVIG, and things like that. So I would recommend doing it for minimum of 2 years. And some patients, they continue both for 4, 5 years, but probably not necessary.
Thank you. Anybody else has any question? Well, so I think there are no more questions in the chat box. Mr. Kalpesh, are there any questions in the chat box? Kalpesh or Nikhil? Fine. So that means end of this webinar. Thank you so much, sir, for that fantastic and lovely talk and updating us on this multiple myeloma update. Thank you so much. Wonderful, fantastic, sir. Good
Thank
to
you, Amit.
and a good
Thank you,
learning,
Dr.
sir.
Agarwal, and all of you. Very lovely experience. Take care. Have a nice weekend.
Thank you so much, sir. Yeah.